Hello and welcome back to Beyond Networks, the evolution of living systems. We've come to a crucial point in this course where we're going to wrap up the first part, which was all about the organism and how to understand it, and move on to the second part where we will use the sort of insights we had in the first part to see whether there are consequences of what we've learned for evolution. And we will see that there are very big connections. So to wrap up this module, let's go back to the very beginning, the lecture about the cell, and how the structure, the fundamental nature, this transient, constantly changing architecture structure of the cell is posing a really big challenge to mechanistic decomposition. Basically, it's impossible to decompose a living cell, an organism, for several reasons. So it's impossible specifically to decompose it into a mechanism. For example, a network representation of the system with individual components represented by nodes and their individual clearly specified interactions represented by edges of the mechanism. One reason for this is that regulatory structure, as we've learned, is radically transient and fluid. It's constantly changing. Its components are changing. They self-assemble, they self-organize. Of course, they also disassemble. They get degraded, they get turned over all the time. And all of this depends crucially on the surrounding cellular milieu, which is determined by the whole cell, not by any specific component of the system. These sort of components of the network, they arise and they disappear based on underlying stochastic processes, which they not only tolerate, but they harvest. So what we have here is sort of an abstract and sort of fuzzy representation of what's going on. The network picture goes in and out of focus, and it has constantly changing components and interactions. There is no well-defined cellular program as a consequence of this. So operations of cellular components rarely follow a predefined sequence of steps. What a cell does depends radically on context. Again, a lot of stochasticity. The coherence only arises out of this random sort of, back and forth of interactions beneath it. And the last point here is that, and that's also very important, you cannot stop the operation of a living system. You cannot stop it like a machine and take it apart and put it together again and press the on button again, okay? Because life is defined by its activity. And also because of the impredicativity of a living system. Basically, there is this dialectic co-generation of the causal components, the processes that constitute it. We've talked about this the last time. So basically, these causes, these processes generate, necessitate, constitute each other. And you cannot just switch them off. They have to be constantly going for life to be going on. So continuous activity is a necessary condition for the existence of an organism, for it to be alive. A corpse is not an organism anymore. So these are serious problems, but they only affect the decomposition of the system. There are also problems that affect the recomposition. I said that about 2 lectures ago, and now I want to come back to this. Okay, so there is a problem with recomposing an entire living system using the tools that we have. For example, the tools of dynamical systems theory that we've introduced in Module 6. So let's dive into that. And for this, we need to go pretty far into the sort of abstract historical and also abstract sort of thinking about how we explain, how we model in science, in physics and in biology. And what I'm going to tell you about is from a wonderful book by theoretical physicist Lee Smolin. It's called Time Reborn. I read this book completely different context than this lecture because I'm interested in the nature of time and change. This is a separate project that I have, but it contains a jewel, and this is an account of how we model in science. And Smolin calls this the Newtonian paradigm, or a bit more informally, doing physics in a box. And it's a very simple and clear description of what dynamical systems theory does. So just to reiterate this, historically, our dynamical systems modeling tools come from physics. They were developed in the context of classical mechanics. Newton was studying planetary motions and the dynamics of the solar system. And so this is a very simple system. And in space, you can delineate, the components of your system fairly well. There's a very famous problem, though, the three-body problem, which means that if you consider more than two bodies in the solar system in a model, you cannot precisely predict the future of the trajectories. So it's not as simple as it sounds. But basically, modeling planetary motions is infinitely simpler than trying to model a living system. The problems we have to solve in biology to understand life are really, really hard. If you understood anything about the last three lectures, I hope it was this. It's incredibly complicated and also incredibly counterintuitive. The organization of life is something amazingly fascinating, mysterious. We still don't know much about it, and extremely hard to understand. It takes work to understand it. Okay, it's also completely different from a normal sort of standard physical system like the solar system. It has all these sort of recursions and impredicativities and all that. So are these tools that we imported from physics really adequate for what we do in biology? Can you just transfer these methods from physics into biology? So let's think very basically about what we're doing when we're making a dynamical systems model, or even broader, when we do experiments. Okay, when we do experiments or we model anything, the first thing we do, and we covered this in the module about systems, module 4, the first thing we do is we artificially mark off and isolate a phenomenon from the continual whirl of the universe, as Smolin writes, is beautifully expressed, okay? Remember Herbert Simon, his near decomposability. He says everything in the universe is connected to everything else, but a lot of these interactions are not very big. But there's a lot of them, so it adds up. So if you isolate a system, isolating a system is an abstraction, a simplification, but you need to do this to apply mathematics, to formalize an actual physical system. So otherwise, you can't get a handle on it. We need to first isolate it from its environment. If you don't do this, we have no modeling tools at all. And this is a big problem, as we will see. Okay, so this sort of separation of the system and its environment, as we've seen before in this lecture, is always, maybe not arbitrary, but definitely depending on the sort of question that we have. There is no natural way for most systems to define their boundaries. Remember the gap gene systems? I completely left out patterning in the head. It was convenient to do so, and also because the pattern process in the head is fairly independent of the patterning process in the trunk region of the embryo. But there are interactions that I simply ignored. So it's always a sort of a difficult question, how to draw the boundary around the system. And also, that's very important. You need to draw this boundary before you can model it or do experiments on it. Otherwise, you don't have a controlled enough situation. Isolated formal systems, and that's very important, are, as Smolin writes, a crude approximation of the real world. We manage to decrease but not eliminate outside influences on the system, and we can never be sure that those outside influences are not actually crucial, important to explain the phenomena that we're studying. So by separating a system from the rest of the universe, I've mentioned this right at the beginning when we discussed the pendulum. It is an absolutely necessary step in modeling nowadays, but it's absolutely artificial. No system is isolated from the rest of the universe. Of course, Smolin writes in the context of modeling the whole universe, which is different. So presumably, there is nothing outside the universe. But let's not get into that, because that's beside the point here. Okay, so then the second step that you do is you define a configuration of the system. So we've drawn the boundaries, and what we're doing now is we're basically defining what the components are that we want to look at. And so the configuration of a system, and I'm slightly modifying Smolin's classical mechanics view here, is, in my view, a list of all the variables and parameters of the system that we need to measure in order to determine everything we want to know about that system. And remember the pendulum again. So we needed to delimit what the pendulum is. We had a description of it, and then we needed to measure the length of the rod and the weight of the bob, which were the parameters of the system. And then, in order to understand how it behaves, we need to know what to measure. So we needed to understand that we can measure the velocity and the position of the bob. So these four numbers together are the configuration space of a pendulum. And of course, for genetic regulatory networks, it's the interconnectivity matrix, some other parameters, and the concentrations of the gene products. Okay, but what's important here is that if we have defined the configuration of the system, we can generalize this to what we call configuration space. This is an abstract space that contains the set of all possible configurations. We've encountered this space when we talked about bifurcation. So bifurcation sets live in this space, and also the Thom's catastrophes, these beautiful shapes that we looked at when we talked about Thom's theory. They live in configuration space, which is the set of all possible configurations. And in a high dimensional system, of course, there are many, many, many possible configurations. But configuration space has a few attributes that are, worth looking at in a bit more detail. So what we need to know is that configuration space as defined is, again, like the boundaries of the system itself, it's a crude approximation to a deeper, more complete description of the problem. We're idealizing specifically on those parts of the system and their interactions that we're interested in, because we're using the models as tools and not as a sort of a general accurate description of the system. Otherwise, we just replace the natural system with a computational system and we won't understand it. So we deliberately focus in and sort of idealize and abstract away all the sort of components, factors, all the surrounding elements that we don't need for a causal explanation of the phenomenon that we're looking at. Okay, so so often a very sort of simple abstract approximation is better than an accurate representation. Remember that from our talk about using models as tools, formal models, of course. So they're very helpful abstractions, okay, for doing what Smolin calls physics in a box. And it should be clear what he means by now. So the basic approach here is that you take a piece of reality, you isolate it from the rest of the universe, and then you sort of focus within that box that you've drawn around the problem. You focus in on those few sort of quantities, entities, processes that you need to explain your phenomena. So you get a very abstract and simplified version of reality here, okay? And this is the biggest problem. Configuration space is timeless. All possible configurations are predefined. They live somewhere in this space. And as you simulate the model, as it changes through changing its parameters, it just moves through this predefined space. This is a very, very, very important concept. We'll revisit it pretty much straight away in the next module when we talk about evolution. It is important to think, are all the possible configuration of evolution predefined? Are they all out there in a massive, super high dimensional space somewhere? How do you get novelty with such a system? Okay, so this description is limited in a way because it doesn't allow the configuration space to change. There are no new configurations that are allowed to come into being. And so we've abstracted away, although we do dynamical systems modeling, we've abstracted away from time. And so this is the central topic of Smolin's book here, when he talks about how doing physics in a box is not possible when you look at the entire universe. There is no box anymore. So what Smolin writes in this context is that Newton adopted the physics in a box approach for the whole universe and everything in it. So since the scientific revolution, and especially Newton's revolution, differential calculus, classical mechanics we think we can describe everything in the universe using this Newtonian paradigm because he said we could. It all goes back to Newton. Okay, and so this approach can be summarized in 3 simple steps. To trace a trajectory of the system. And remember Waddington's landscape, we had these developmental trajectories that we wanted to explain. So to define such a trajectory, you have to specify 3 things. 1, that's the initial condition of the system. You need to know a little bit of, not the history, but at least the present state of the system for being able to define the future of the system. Okay, so the boundary conditions of the system, and that's the box. That's the second thing we need to define, okay? Where and when does the system apply? Not only boundaries in space, but also in time, of course. Okay, so systems are all spatio-temporally bounded. So we draw the box around. And so what's important here is that we impose these conditions, both the initial and the boundary conditions, before we even start simulating or solving the equations. And we also need to provide, before we model, the rules of change, which are encoded, usually described by and encoded in equations. For example, differential equations in a dynamical systems model. So the rules usually don't change. I told you a little bit about time dependence and how the parameters of certain systems can change, so you can get bifurcations over time, but usually the basic form of the equation stays the same. And actually dynamical systems theory doesn't allow you to just rewrite the rules. Okay, so these are things that are given from outside, pre-given before you even model your system. So you need to impose them from the outside, okay? And the second sort of big problem here is there is no true emergence. What do I mean by true emergence? I haven't even introduced this term so far. What I mean by true emergence here is that in the universe, sometimes there are events that bring new rules into play. New configurations come up. We will talk a lot about this when we talk about evolution. Think about the origin of life. As soon as life originates, the organization of the living appears for the first time. This is a certain type of organization that has not existed before. It is not covered by the laws of chemistry and physics that describe a lifeless universe. This is not vitalism, but it just means that when life appears, the universe becomes enriched. There are new rules added to those that already exist at that point. And the same may apply for, of course, for the emergence of culture and consciousness later on in evolution. There are actually more such events that you could possibly define. So what I'm talking about here is not an emergence in the sense of a pattern emerging from a bunch of equations. We'll talk about that in a second. But for a stronger sense of emergence in that there are really new rules and a new level of organization and a new level of explanation that you need to understand that new level of organization. So you can think about it in a Wim Satkin sense as a new level of organization arising. Now you cannot model that sort of event using the Newtonian paradigm or physics in a box because of its pre-given conditions that are too inflexible to allow for this to happen. So we have a problem here, right? So how does this apply to biology? Smolin writes about physics, and I want to sort of diverge a little bit right now and present a quite detailed example of how we use the Newtonian paradigm to model morphogenetic fields that are important in pattern formation and development. So we're going to do this in parallel examining, always thinking back to Smolin's argument. And I'm going to show you how the way we model systems in biology right now fits perfectly into this physics in a box paradigm that Smolin is describing. And the classic sort of modeling that I'm going to introduce, which I have absolutely dreadfully neglected so far when I talked about different forms of theory, is that of Turing patterning systems. These are very, very famous models in pattern formation and morphogenesis. They go back to a very, very famous, one of the most highly cited papers by Alan Turing, the father of the modern computer, who came up with the Turing test, the Turing machine. And so he wrote one single paper about this problem, which was completely outside his own field. He was thinking about how do biological patterns arise. And he broke with a paradigm at the time. He completely destroyed a very general idea, a conviction that people had, and that was that diffusion always equalizes things. So his paper, which had one figure and just 5 or 6, I can't remember, references, those were the times. This is the figure, it's like a hand-drawn, looks like the coat of a cow. We'll see why in a minute. And so in this paper, the main insight that Turing had, he studies the principles of morphogenesis, and he came up with a system that could make such inhomogeneous patterns, black and white pattern, from a homogeneous initial condition. So the initial condition here was just white or black, doesn't matter. Okay, the box is quite literally a box, a field, a spatial domain in which the pattern occurs. And the rules are two substances that interact and diffuse at the same time. Okay, so the main point of this paper is that diffusion can not only homogenize the distribution of a chemical. I'm sitting in this room hoping that diffusion is homogenizing the concentration of air molecules in the room, right? This is what diffusion has been known to do for a long time. But diffusion can also generate pattern from perturbation of a homogeneous initial state. So you have a homogeneous initial state, you have some fluctuations. And from those fluctuations, a pattern, a coherent pattern arises. This is beautiful because, so this is a perfect, it's a very powerful modeling paradigm, and it reflects a lot of the arguments I've made about, for example, harvesting fluctuations for coherent behavior. That's what this system is doing. Okay, so it's great, fantastic. So it seems very suitable to study biological systems so far. A little aside, look at the citation count of this paper. When Turing published it in 1952, it went for 20 years, more or less uncited. I think the first 20 years it got 17 citations. And then in the early 70s, theoretical biology takes off a little bit. There's a bunch of models. There's the work by Gier and Meinhardt that I'm going to show you in a minute that uses Turing's formalism, and then systems biology kicks off at, in the 90s, and then here in the 2000s. And now this paper has hundreds of citations. This is, by the way, a graph that was provided by my friend and collaborator, Lee Altenberg. And it shows you, a paper like this is called a Sleeping Beauty paper. It shows you the best theory. The best theory is decades ahead of its time, and then suddenly becomes inspiring at a much later date. Just as a very brief aside, this doesn't work anymore today. If you publish papers like that, you're dead. You don't have a career, because we have this stupid, short-sighted sort of academic system nowadays that's sort of based on very short-term sort of productivity. Completely stupid for theoreticians. That's why good theoreticians are threatened by extinction in academia nowadays. But we'll get back to that when we talk about bullshit in science in a future module. So let's not get distracted here. One particular instance, so Turing's model was, I'm not going to talk about it in detail because it was slightly bizarre. It was a linear system, and it had certain other properties that were not very realistic. But in the early 70s, Gier and Meinhardt came up independently of Turing with a specific instantiation of the system, and then later discovered that Turing had already published it 2 decades earlier. So this particular instance of a Turing system is called a local self-activation with lateral inhibition system, or LALI system. It consists of 2 components, an activator and an inhibitor, remember? And they diffuse, these substances diffuse. So the activator activates itself, but also the inhibitor, and the inhibitor diffuses away faster than the activator and inhibits the activator. So here we have a bit of a paradox. So an activator activates an inhibitor, it's a negative feedback loop, and what do you get from that? Okay, so in the best case, again, so we're defining here an initial condition, it's homogeneous, you can see it here, concentration of the two factors. The dots here indicate a certain level of noise, okay? So there are fluctuations. And then you draw the box. So there's a tissue, it's one-dimensional here, it's just a line, okay? Left and right, there's a tissue in the middle. And then you simulate the system according to the rules defined by this little network. And what you get is the activator, because of a fluctuation, will enrich at one end of the tissue more and more and more. And as it enriches, it will activate the inhibitor more and more, but auto-activation of the activator can keep it active here. It can outcompete the inhibitor, while the inhibitor diffuses more quickly than the activator and shuts it down in this area. Basically, activator on here, activator off here, because of diffusion. So unlike the gap gene system, this system is not driven by local genetic interactions, but by diffusion, non-local sort of mechanisms. And we'll come back to that in a second. So that's another reason I chose this example for this particular lecture. You can change the diffusion constants and create more complicated short range patterns like these stripes. And so basically, what's important, what I need to emphasize again, is that the pattern is generated through the difference in diffusion rates between activator and inhibitor. It doesn't depend very closely on the details here. So there's several interaction schemes that will produce very similar patterns, and the Lally interaction scheme that I'm presenting here is just one of them. That's very important. So this goes back again to the right level at which we need to look at the mechanism here of this patterning generator. So these Turing systems can do different things, like any other model I was telling you. So there's a noisy initial condition here. Most of the time it will not do anything. I was telling you for gene circuits already, that's usually the default behavior. Nothing exciting happens. Everything just dies out or remains uniform. You can have temporal oscillations. You can have very short range stationary waves, sort of salt and pepper patterns that oscillate sometimes. That's like the gray noise on your TV. These are all not very interesting. But then you have two biologically interesting cases of pattern that are happening here. One is it can generate oscillatory traveling waves. It is a dissipative system, just like the Belousov-Zhabotinsky reaction I was showing you last time. And it can produce stationary waves, or if you go to two dimensions, it can produce rolling spirals like these, or that's the same mode as the traveling wave here, or these stripes and dots. Very versatile pattern generator. And what's really interesting here is that this can be considered a mechanism of pattern formation, but not all its components, its functional components, are localized. Diffusion is a non-local sort of factor in this patterning process. So a bit of a headache for mechanistic explanation, it seems. Here is a beautiful example, an early example of a Turing model from the early 80s, where Jim Murray, famous theoretical biologist at the time, came up with a model that explains different patterns on animal codes. So what you see here are a bit more complicated spatial domains that approximate one of these safari rugs that you can see sometimes in old-fashioned homes still. And he's now simulating patterns, code patterns on this sort of realistic topology, geometry of the spatial domain. And you have different parameters, and you get different, you know, very sort of long range or very sort of fine grained spotty and stripy patterns with more or less black and white here. Okay? So beautiful. Have you ever looked at a tapir in a zoo and realized it's a walking Turing system? When they're little, they have these dots and stripes. I thank my friend Nick Monk for this example. And then when they grow up, the parameters of the Turing system are changing. You get a bifurcation during development from this stripy pattern to a different Turing mode, it's called, a much more long-range mode, where you get this beautiful long-range pattern here in the mother. So the other thing you can do is you can, instead of changing the rate of diffusion, you can change the size of the domain. Okay, and this brings me to an interesting point. The pattern that is generated here depends on the geometry of the system. The most beautiful example of this is a recent publication by Dillon and colleagues in the Bulletin of Mathematical Biology. And these people used the geographic information system to scan the shape and the size of different realistic shapes and sizes of different species of gecko. And so these little lizards, these representations are realistic gecko shapes in the computer. And then they simulate their Turing systems on those shapes. And they can show that the size variation and the shape variation in these species can account for much of the patterning differences between the species. Why is this so interesting? Okay, if you think about it in mechanistic decomposition terms, we've gone a big step here from just doing genetics and causal modeling to recomposition of the system, including this causal factor here that is a global property of the system. So where does the shape of the animal come from? From the animal itself. Of course, it is generated by the animal, by its growth, by its developmental processes. But what we do when we model this is we plug it into the computer, we measure it outside, and we put it in as a boundary condition. The shape of the domain is a boundary condition for the system. It's a constraint to the system. So the pattern not only depends on local interactions between the activator and the inhibitor, but also on the global shape of the domain to be patterned. So what you see here is a type of morphogenetic field. Remember that? So it's a beautiful example of a morphogenetic field where the pattern does not only depend, there's a global order. The pattern is a global pattern. It not only depends on local interactions, but it also depends crucially on the shape of the domain on which it is happening. So although this is not a localized component, the geometry of the lizard, it's not a localized component in the strict sense. It is a perfectly acceptable Woodwardian cause for two reasons. First of all, if you change the shape of the lizard, I don't want to think about how you do that, but you can do it. Then you will have a different pattern, okay? That's one sense in which it is interventionist. The other sense goes back to my argument that I've shown you before. Boundary conditions are constraints, and I've argued before and I've showed you that constraints are perfect Woodwardian causes. There's no problem with them. They go between levels, but there is no problem that this is a system-wide parameter. Okay, so not all causes need to be localized for a causal explanation to apply. This is one of the reasons why it's so important to recompose the system. This is one of the limitations you run into if you only do genetics. You cannot explain this pattern at all. But what we've done here is we've cheated, of course, when we model the system, because we imposed the model itself does not explain the shape of the lizard at all. It just assumes it from outside. So we've drawn an artificial box, not just in terms of defining the structure, the geometry of the lizard, but we are not considering where the shape of the lizard comes from. So we've excluded that and thrown it into the environment of the system, into the rest of the universe. And of course, if you truly want to understand the patterns, you would have to understand the entire shape of the lizard. Turing apparently remarkably commented about zebras. He said the stripes are easy, but what about the horse part? So here, the horse part is missing. Okay, we cannot explain with this model the way the horse looks like or the lizard looks like in this case, but we just impose it from outside. Okay, and this is a serious limitation of our modeling approaches. So these sort of modeling approaches, dynamical systems models, Turing systems, take us a far step from pure decomposition of a mechanism, which doesn't work because the components are not localizable, to recomposition of the system, and the mechanistic explanation of a morphogenetic field where not all the causal factors are localized. So this is a big step forward. It's a step towards a processual view of development. It's a step towards a global systems-wide view of development. But does it go far enough to explain the behavior of an entire living system? And here's the answer. Rosen, one of his main points in his book, Robert Rosen, has a very, very difficult to understand mathematical proof using category theory, where he argues that this type of organization that he shows in this diagram with closure to efficient causation, because living systems have this type of organization, they fall into a different category than those systems for which we can have mechanistic explanations. He calls that simulation. Okay, so simulation here is dynamical systems modeling, more or less. Okay, so he says you cannot use dynamical systems modeling because it only applies to categories of systems to which those living systems with closure to efficient causation do not belong. Okay, there's a very formal proof. It's been very controversial, but it has not been refuted yet. It stands as it is right now. So this is very abstract, and I don't want to go into the details of the proof and its controversies. So instead, let's try and get an intuitive feeling of the argument here. And so what I'll do is I'll use Janny Hofmeier's more detailed mapping of Rosens' scheme onto the cell to make this argument. So this is his very complicated scheme of the hierarchical cycle that forms the self-fabricating organization of the cells. Just a quick reminder, there's 3 efficient causes, enzymes and ribosomes. They mediate all of the covalent biochemistry and chemistry in the cell. The intracellular milieu allows for folding, self-assembly of proteins into functional proteins and protein assemblies or riboprotein assemblies like ribosomes. And then the transporters that are also proteins themselves are the efficient cause for the intracellular milieu by pumping out or letting in specific ions, for example, and regulating the pH of the cells, et cetera, et cetera, et cetera. These are the 3 efficient causes. They stand in a circular relationship, just like Rosens' abstract scheme. We've been through this. So now let's have a look at how we would model this using dynamical systems theory, right? So in the Newtonian approach that we're usually taking and that we've taken, I hope you've noticed when we did the Turing model, we did a perfectly Newtonian thing. We defined the initial conditions, we defined the boundary conditions, we defined the rules. There was a predefined configuration space, everything kosher there, no problem, okay? Everything perfectly according to the Newtonian paradigm. So what we need to do is specify initial conditions, boundary conditions, and the rules of change for a cell model here. Let's look at that. And let's start at the bottom, number 3, the rules of change. So we've seen before, 3 lectures ago, that, and also again with the work constraint cycles in last lecture, that so living systems have this recursive structure, okay? The rules need to constantly be changing. You need basically a system of equations that constantly rewrites itself. And that's not possible using differential calculus. Okay, that's just not what it was designed to do. But there is a way out. You can use different mathematical formalisms. I don't want to go into detail, but there's a beautiful paper by Matteo Mossio and colleagues from 2009, Giuseppe Lomo is on this as well, where they explain that if you have a mathematical formalism that allows you to rewrite the rules based on its own operation, then you can overcome this problem of recursion. Okay, and so one of these formalisms is called lambda calculus. You're free to look this up. I can't go into details right here, but I just want to tell you that there is a formalism. It's harder to analyze than dynamical systems, but you can use it. A mathematical formalism exists, and you can overcome this problem. But as even those authors admit in their paper, there are additional problems. Okay, so the next one is the boundary conditions, the box, right? And so Smolin was criticizing this physics in a box, talking about the entire universe. There is no box anymore, okay? So everything is in the box if you want to explain the whole universe. With organisms, there's a similar problem almost, because, I should have mentioned here below, sorry for this. The configuration space is co-evolving with the system when the rules rewrite themselves, okay? We'll come back to that in a sec. So back to the box. So if you have a closure of constraints, the boundary conditions, Remember, the constraints are boundary conditions. These boundary conditions get weirdly wrapped into the system. So Rosens' trick of accounting for all the efficient causes from within the system, they mean that all those boundary conditions that you need for life to continue need to be generated from within the system. But that's not what a boundary condition is in dynamical systems theory. Okay, you can no longer draw the boundary around an organism before you start modeling it. It has to draw itself. Okay, organisms draw their own boundaries by this work constraint cycle, by furthering the closure of constraints, by continuing the closure of constraints. Okay, so philosopher James DeFrisco and Matteo Mosio have called this organizational continuity. This is a paper that's not really published yet, so I shouldn't talk too much about it, but it's coming out very soon. Okay, so this is very interesting. So you maintain this closure, right? And by doing this, you draw your own box. So you cannot specify the box in which an organism exists before you start modeling it, okay? That's a problem. That's a headache. Could be that you can overcome that as well with lambda calculus, but I'm not sure. So we'll revisit this problem, and it has a really interesting consequence. It means that organisms are agents, okay? They have something called immanence. And immanence is the ability to generate action from within. They can modify their own constraints while maintaining closure of constraints by doing these work constraint cycles. Remember? So they have this needful freedom that Hans Jonas was talking about it. They're autonomous, in Alvaro Moreno's term, from their environment in some sense, okay? So this will keep us busy when we think about evolution. This has large consequences for an evolutionary theory. But in this present context, can we have a mechanistic explanation of the whole organism? This is the killer. So the initial conditions, we don't know how to formulate the initial conditions because of impredicativity. These different efficient causes in organisms, they generate themselves, each other, all the time. And they need to be continuing. To be alive is to be active. Remember, you cannot stop life, take it apart, understand it, and put it together again. So you need to understand basically the continuous flowing history of life since its origin. And this origin of life is a huge mystery today. How this sort of impredicative dynamic, this dialectic between different efficient causes came into being is a really big question that we don't understand very well right now. But once it got going, it's been going ever since. It's an amazing thing to think about. Your metabolism, your digestion, basically, you know, has been going on since the beginning of life. It's never stopped. I mean, not literally the digestion, but the cellular metabolism of at least your germline. It's amazing. You're continuous with the first living cell in this sense. It's amazing, absolutely amazing sort of insight if you think about it deeply enough. Okay, but it's a huge riddle. And these tools that we have for modeling that come from physics, they are not able to deal with impredicative systems. In fact, impredicative systems are a huge problem. That's why a lot of people work on them. They're a problem. Okay, we don't know how to deal with them at all. So there's a fundamental problem here. And so what we're left with is this need to move beyond mechanism without becoming vitalists, of course, and saying, okay, it's all just a mystery. How can we move forward? Because this very powerful approach of dynamic mechanistic explanation with decomposition using genetics, causal modeling, and recomposition using dynamical systems modeling. So we've just seen that this is a powerful and sort of really amazing approach to study morphogenetic fields. Especially recomposition is crucial because there are non-local causes in biological systems. So to get a processual view of development and how the organism forms, this is crucial and it's very powerful. But if you want to understand the entire organism, okay, you have to move beyond this biology in a box approach. So you can explain individual morphogenetic fields as long as you want with biology in a box. You draw a box around it, you can explain a lot about it, but it falls woefully short of understanding life itself. And the problem here is not that we just need more data. All these people that are just going about getting new data sets, bigger data, machine learning, blah, 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 and then at some point, the secret of life will pop out of this. That's just ignorant. I'm sorry. What we're missing here is the point. The point is that we don't even have the methods or the concepts to think about what life is. We're just beginning to develop those. And so what we need here are new ideas. We need new mathematical approaches to deal with this. And we need to take the philosophical problems with the organization of life really, really serious if we want to understand life itself. And this should be what biology is doing. So François Jacot said, we no longer study life in the laboratory. In his La Logique du Vivant, 1970, and he was happy about this. But this is the whole point of biology, to understand life. Why is nobody paying attention to this? It's not an unresolvable problem, but if nobody's trying to work on it, or nobody's allowed to work on it, or get financed to work on it, then we're not going to solve it, of course. This is a grand challenge for biology that lays by the wayside because nobody is working on it, because we're no longer allowed to do this. Instead, we're gathering more data, and we're just getting more and more confused. And this is why, because we don't understand what life is. So we need to get a grip on that. We need to refocus on what our field is really about and has always been about. Remember, systems biology, it's just the last 100 years that we're an aberration, where we focused in on mechanisms, on molecules. It's time to get back and put the organism at the center of biology again. So this was the first part of this lecture. The second part will be shorter, 5 modules. And what we're going to do is, so what I've presented here was sort of a continuous argument to show you that we have amazing tools to look at biological systems, but they're nowhere near adequate in light of the amazingly fascinating and mysterious and complicated nature of life. But what I've also shown you here is that the organization of life implies a few things. For example, the agency of organisms. Organisms do stuff. They want stuff. They have goals. And so we'll have to go back and ask, don't we have to deal with this observable fact when we think about the evolution, the interaction of organisms during their ecological interactions or during their evolutionary history? What kind of consequences does this systems thinking, taken serious, like we do here, have for evolutionary theory? Where could we even start to think about the consequences this has? And so we'll come to a view that we need not just a new sort of approach to study living systems, but we also need a new kind of theory of evolution. It's not that the old theory is wrong, it's just limited. To go beyond its limitations, we will need to develop an agent-based view of evolution. And what that means, I'll try to explain over the next 5 modules. I hope you tune in again. Thanks for listening and for coming on this rather strenuous journey, conceptual journey through the theory of the organism. I hope you learned something. I hope you see living beings in a different light with more respect as the beautifully complicated and mysterious systems they are. Thank you very much. See you next time.